Welcome friends. Today we are going to discuss about the difference between prokaryotic cells and eukaryotic cells. So the first point of difference is diameter. So this prokaryotic cells, they are very small. Their diameter is about 1 μm. Okay, and eukaryotic cells have diameter of about 10 to 100 μm. Next is cell wall. So these prokaryotic cells, they are mainly non-cellulosic, but some bacteria, they have cell wall. Okay. Next is eukaryotic cell. They are cellulosic in case of fungal cells and plant cells. Okay. Next is cytoskeleton. The cytoskeleton, which provides a shape to the cell, they are absent in prokaryote, but they are present in eukaryote. Fourth point of difference is nucleolus. These nucleolus, they are absent because prokaryotic cells, they do not have true nucleus. Nucleolus is absent but it is present in eukaryotic cells. We all know that the chromatin material is present in the cytoplasm. There is no nuclear envelope. There is no nucleus in prokaryotic cell. Okay, so nuclear envelope is also absent in prokaryote. It is present in eukaryote. So the main point of difference between prokaryote and eukaryotic cells are eukaryotic cells have true nucleus. I'm highlighting this is because this is the main point of difference. Next is other cell organelles like mitochondria, chloroplast, endomembrane system. All these are absent in prokaryotes and they are present in eukaryotes. Next is chromatin. So these chromatin, they are single in prokaryotes and multiple in eukaryotes. These chromatin, they are non-histone and they are circular. Did you understand me? They are single non-histone, no histone proteins are present and circular DNA. And in eukaryotes, there are multiple chromatin which are associated with histone. Associated with histone as well as with other proteins as well as other proteins. And also the important point that they are linear. Eukaryotic DNA are linear and prokaryotic is circular. Next one is DNA. So prokaryotic DNA, mostly they are non-repetitive. Mostly non-repetitive. So do you remember by using which curve we can analyze whether the DNA is repetitive or non-repetitive? Can you recall and write me in the comment box? Okay, so next is eukaryotic DNA. They are mostly repetitive in nature. Next is DNA content in base pair. So the DNA content in base pair for prokaryote will range about from 1 into 10 to the power 6 to about 5 into 10 to the power 6 base pair. And for eukaryotes, it's 1.5 into 10 to the power 7 to about 6 into 10 to the power 9 base pairs. Okay, so the DNA length is bigger, so that's why the number of bases will also be more simple concept. So the ribosomes, they are of primitive 70S type and in eukaryotes it's of 80S type. Now please learn that what are the two subunits of 70S type and what are the two subunits of 80S type. Okay, please read it. Next is exocytosis and endocytosis. They are absent in prokaryote but present in eukaryote. Exocytosis is when something leaves the cell, endocytosis when something enters the cell. Next is gene. So these genes are very important point. They are continuous in prokaryotes. And introns are absent. I write it with different color that introns are absent in prokaryotes. Okay, say and in eukaryotes these genes they are interrupted. Okay, interrupted because introns are present in between the exons. Okay, and there is no collinearity. Collinearity is not maintained in eukaryotes. But in prokaryote, collinearity will be maintained. That means that means collinearity in between the sequence of nucleotide in the DNA and the sequence of amino acid in the polypeptide is maintained. Okay, so sequence of nucleotide in DNA and sequence of amino acid in polypeptide, this is maintained in prokaryotes. So we'll discuss in detail when we will be doing the topics of fundamentals. Next is replicon. So these replicon, they are the origin of replication. Okay, they are the origin of replication and they are single in prokaryotes and there are multiple in eukaryotes. Okay, the replicon process is complex in eukaryotes due to the presence of some associated proteins forming nucleosome. Okay, next is telomerase. This is absent in prokaryotes and present in eukaryotes. We will discuss all about these in detail. I'm just comparing, so I'm not going into much detail. We will discuss about all these in detail when I'll be discussing with you the topic of fundamentals. Next is DNA polymerase. In prokaryotes, the DNA polymerase they are of three types. What they are? Type 1, 2, 3, which is 1, 2, and 3. But in eukaryotes, the DNA polymerase they are of six types. What they are? Alpha, beta, delta, epsilon, gamma, and this last letter is pronounced as xi. Okay, this is xi, something like it looks like E. Okay, said to be as E. So I have written it clearly for you, okay, by pausing the video. Next is RNA polymerase. This RNA polymerase is of only one type in prokaryote and it is of three types in eukaryotes. So it is of three types in eukaryotes. What they are? So number one is, so three types of RNA polymerase are required for eukaryotic cells. Number one is polymerase 1. Polymerase 1 helps in the synthesis of rRNA, ribosomal RNA. Polymerase 2 helps in the synthesis of mRNA. And small nucleolar RNA. And polymerase 3 helps in the synthesis of tRNA and 5S rRNA. Okay, so most of the rRNA are synthesized by polymerase 1, but there is one exception, that is 5S rRNA, which is synthesized by polymerase 3. Next is genetic recombination. So this genetic recombination, it is generally unidirectional in prokaryotes. But in eukaryotes, it involves reciprocal exchange. that is crossing over, right? Next is mRNA. So these mRNA, they are polycistronic in case of eukaryote. So you can remember prokaryote starts with P, polycistronic starts with P. So in prokaryotic organisms, these mRNA, they are polycistronic. Okay, but in eukaryotes, they are monocistronic. Also, one thing important I'm telling you, that in case of prokaryotic organisms, as you if you remember, recall it, that RNA transcript, in case of prokaryotes, RNA transcript, it is free to associate with ribosomes prior to transcription. Okay, did you understand? Prior to the completion of the transcription, RNA transcript, they are free to associate with ribosomes. But in case of eukaryotes, these RNA transcript, it is not free to associate with ribosomes prior to the completion of transcription. In a simpler language, in case of prokaryotic organisms, before the transcription is complete, the transcript is free to associate with ribosomes. But in eukaryotes, transcription must be complete for the before the RNA transcript associates with ribosome. So until the transcription is complete, RNA transcript cannot associate with ribosome in case of eukaryotes. But in case of prokaryotes, RNA transcript can associate with ribosomes before the transcription is complete. Did you get me? Next is, these RNA maturation is not required by splicing mechanism. Okay, there is no splicing mechanism seen in prokaryotes. But in eukaryotes, maturation of RNA transcript, it is required to form mRNA. So maturation is not required in case of prokaryotes, and maturation of mRNA is required in case of eukaryotes. Next is promoter element for transcription. So I'm using a different color for this. Promoter element for transcription, it is known as box in case of prokaryotes, which is at minus 10 base pair, and Tata box, which the sequence is TT GACA, TTGACA, TT GACA box, Tata box, which is at minus 35 base pair. What about eukaryotes? Eukaryotes also have Tata box, which is at minus 30 base pair. Another is they have the cat box. Where is this located? This is located at minus 70 base pair. Okay. Also, they have GC box, which is at minus 110 base pair. Another point of difference is post-transcriptional modification of RNA. So this is absent in prokaryotes, but in eukaryotes, just after the transcription and before the splicing, these RNAs which are synthesized, they are required to be methylated. So in which stage? After transcription and before splicing. Okay, I am writing it here. After transcription and prior to splicing. What happens? That these RNA transcripts which are synthesized, they are methylated at which end? At 5' end, which is known as capping. Okay, and they are polyadenylated. Polyadenylated means several A residues will be added like A base pairs polyadenylated at 3 prime end, okay, which is known as tail. So this is the post-transcriptional modification of RNA. Next is signal for translation. Signal for translation here is binding of mRNA with small subunit of ribosome. So where this ribosome will bind on mRNA, this sequence has to be searched. So in which region will the ribosome bind to mRNA? In which region will the small subunit of ribosome will bind to mRNA? So in case of prokaryotes, this binding involves a sequence known as Shine-Dalgarno sequence. What is the basis of the sequence? It is AGG. Okay, this is the sequence on which the mRNA and the small subunit associates or binds. This is Shine-Dalgarno sequence. What about eukaryotes? Now, in eukaryotes, surrounding the start site, which is AUG, surrounding AUG, there is a sequence named as Kozak sequence. What is the base of this sequence? It is AUGG. Now this Kozak sequence acts as a signal sequence for binding of small subunit. So I write here signal sequence for binding of small subunit with small subunit of ribosome with mRNA. So in case of prokaryote, Shine-Dalgarno sequence serves as the signal sequence for binding of small subunit with ribosome, and in case of eukaryote, Kozak sequence serves as a signal sequence for binding of small subunit of ribosome with mRNA. Next is initiating tRNA. So initiating tRNA is what in case of prokaryote? It is F-mate tRNA, which is formal methionine tRNA. Also we write as F-met tRNA. Okay, formal methionine tRNA. And in case of eukaryotes, it is simply methionine tRNA. Formal group is not there. Okay, last is inhibition of protein synthesis. So in case of prokaryotes, the antibiotics chloramphenicol, okay, it inhibits protein synthesis by inhibiting bacterial ribosome. And in case of eukaryotes, it is cycloheximide, which inhibits the protein synthesis. So let's revise what we have studied. So diameter, prokaryotic cell are very small. They have diameter of about 1 μm. Eukaryotic cell have a diameter of about 10 to 100 μm. The prokaryotic cell, mostly they are non-cellulosic. Some bacteria, they have cell wall. In eukaryotes, plant and fungal cells, they are cellulosic. Cytoskeleton is absent in prokaryotes, present in eukaryotes. Nucleolus is absent in prokaryote, present in eukaryote. Nuclear envelope is not present in prokaryote but present in eukaryote. Some cytoplasmic organelles like mitochondria, chloroplast, endomembrane system, all these are absent in prokaryotes and present in eukaryotes. About chromatin, the chromatin is single and non-histone and the DNA is circular in nature, but in eukaryotes, the chromatin is multiple and they are associated with histone proteins and some other proteins like DNA fibrils. And the chromatin is linear. Next is DNA. So this DNA are They are non-repetitive in case of prokaryotes, and they are repetitive in case of eukaryotes. Next is DNA content in base pair. It is about 10 to the power 6 to 5 into 10 to the power 6 base pair, but it is very high in case of eukaryotes, which is around 1.5 into 10 to the power 7 to 6 into 10 to the power 9. Okay, just remember the approximate value. Next is ribosome is 70S in prokaryotes, 80S in eukaryotes. Exocytosis and endocytosis kind of process is absent in prokaryotes but present in eukaryotes. Next is gene. These genes are continuous, that is, introns are absent. Okay, only the coding sequences are present in prokaryotes. In case of eukaryotes, the genes are interrupted. Several introns are present in between the exons. The collinearity is not maintained in case of eukaryotes. Next is replicon is single. In case of prokaryotes, replicon is the origin of replication. In eukaryotes, it is multiple. Telomerase is absent in prokaryotes, but it is present in eukaryotes. Now, DNA polymerase is of three types in case of prokaryotes, and it is six types: alpha, beta, delta, epsilon, gamma, and xi. They all are present in eukaryotes. RNA polymerase is of only one type in prokaryote, and it is of three types in eukaryotes. Polymerase 1, which synthesize rRNA, and polymerase 2, which synthesize mRNA, snRNA, which is small nuclear RNA, and polymerase 3 synthesizes tRNA and 5S rRNA. Genetic recombination is unidirectional in prokaryotes and it shows reciprocal exchange in eukaryotes. mRNA is polycistronic in case of prokaryotes and monocistronic in case of eukaryotes. Now, promoter element for transcription, this Pribno box and Tata box in case of prokaryotes, and Tata box, Cat box, and GC box in case of eukaryotes. Next is post-transcriptional modification of rRNA. It is absent in prokaryotes, but before splicing, 5' cap and uh, 3' polyadenylation is seen in eukaryotes. Now, signal for translation, Shine-Dalgarno sequence is the signal where small subunit of ribosome binds to mRNA. But in eukaryotes, it is a Kozak sequence which is near to AUG, which is the start codon. Near to AUG, the Kozak sequence is present where the small subunit of ribosome binds in case of eukaryotes. Initiating tRNA is formal methionine tRNA in case of prokaryotes, and is simply methionine tRNA in case of eukaryotes. Last is inhibition of protein synthesis. Chloramphenicol inhibits prokaryotic protein synthesis, and cycloheximide inhibits eukaryotic protein synthesis. So that's it. If this video has helped you, please hit the like button and subscribe to the channel. Thank you so much.